already?
It's difficult drawing. Scientific drawing is difficult.
Okay. Are you all set? You're recording? All right, so I guess I'll start up now. My name is Frank Vázquez. Hi.
Hi, Frank.
Hi.
And what I'm going to be talking today about lipids. And really what I'm going to be trying to, what I'm going to be trying to do, at least that was kind of what I was trying to do with these slides, is we're going to try and understand this complicated picture right here, this cell that has a whole bunch of stuff going on, and we're going to try and understand it in terms of the chemistry that we can use to understand salad dressing. And so we're going to try and really simplify our picture of how the cell is able to form these walls and use and why certain molecules have certain chemical characteristics inside the cell and what kind of purpose they serve. But instead of trying to really jump into the biology and understand the biology, really what we want to understand is, why does this salad dressing have two different layers? What stuff is in the bottom layer? What stuff is in the top layer? Why is that stuff in the bottom layer? Why is that stuff in the top layer? How can we mix up this salad dressing so it's one liquid instead of these two layers? And how can we use that information to understand the cell? That's going to be the whole point of all these slides.
Any cell?
Yeah,
Any
any
cell?
cell.
Any cell? Okay. Sorry. Just
And I guess not the cell
wanted to
membrane,
qualify. Okay. Yeah.
more so than the cell itself. But pretty much, if you understand the cell membrane, I would say you understand a lot about the cell.
Okay.
And the tools that we're going to start off with, we're not just going to jump into that. First, we're going to kind of set up a toolbox so that we can understand essentially the salad dressing, which is we're going to talk about electronegativity, which I think has been covered already, but we'll just go over it again. And polarity, what it means for a molecule to be polar, and hydrophobic versus hydrophilic molecules. And we're really, this line here, I should have highlighted it, is the most important line. Is like dissolves like. Molecules that are similar dissolve each other. And molecules that are not similar do not dissolve each other. And then I'm going to talk about amphiphiles, which I'll define later so you don't have to worry about what that is. So just kind of a brief refresher on electronegativity. If we look at the periodic table, you can see how it's colored here. This region is colored bluer and this region is colored more red. The reason it's colored that way is that the more red elements are called electronegative, which just means that they're going to attract more electrons to each other. So if you have some sort of a molecule, draw. So if we have some sort of molecule that has, actually this is kind of too late. Let's say we have this molecule right here, so this would be ethanol. And if we put one of these electronegative elements, specifically oxygen in this case, on the molecule, what's going to happen is that all of the electrons that are shared by all of these atoms are going to tend to be attracted to this region right here. The oxygen pulls the electrons towards itself, which means that you're going to have this end be a bit more negative and this end be a bit more positive. So you're essentially going to have something that's more like a magnet, whereas if you have the same molecule without the OH on it, there's nothing pulling there's nothing really pulling electrons in any one given direction. The electrons are shared among the whole thing, so you don't have this separation of charge between the two. You don't have one end that's going to be more negative and one end that's going to be more positive. Oh, that means I was on the camera. And kind of more in depth here with more of a real world example. This polarity is what we call this process. When we have some sort of element that is drawing electrons towards itself and it creates that separation that I showed over there, we say that the molecule is polar. If a molecule does not have that, does not have an element that's going to be more electronegative and pull electrons towards it, we say it's nonpolar. So 2 examples, we have this 3-carbon molecule here, that's propane, so cooking gas. I don't think it's the gas that comes in through for your stove. I think this would be more for like a barbecue. And then we have isopropanol, which is rubbing alcohol. So this same molecule with an oxygen placed right here. And if you start thinking about what these molecules are like in real life, you can already see what a big difference putting that oxygen makes. Propane is a gas. Propane is very different, it smells different, it looks different, it has completely different properties than rubbing alcohol. And to kind of push this idea some more, so once again, what does it mean to be a polar molecule? Essentially it means you're hydrophilic, you're a water-loving molecule. And what that means is that the molecule is soluble in water. Water is such an important molecule that we choose to define different molecules in terms of whether they're soluble in water and whether they're not soluble in water. And we name them accordingly. So hydrophilic, water-loving, and we can ask again, why is this the case? What's going on here? What we have is that we have this oxygen, and it pulls all those electrons towards itself and essentially acts like a magnet. So you're going to have a positive end and a negative end, and the positive end is going to be attracted to the negative end. And this is going to have huge ramifications for how these different molecules react when they're in water, but also how water can order itself around them too. You can see here how I'm kind of alluding to it the way I drew this. I didn't stick the red oxygen next to this oxygen here, because that's not the way it would be in a real liquid. They're going to arrange themselves so that the positive ends are always towards the negative ends, and that's going to lead to the actual structure that water has. Even when it's a liquid, water is going to have some sort of structure associated with it. And when it's an ice, it has a very specific structure that's based off of how the positive hydrogens arrange to meet up with the negative oxygens, just like you would have in a magnet. By the way, if there's any questions, feel free to interrupt.
Yeah, just to clarify, so with that water molecule right there, it's one of the positive hydrogens that's attracted to hooking up with the negative oxygen over there, and does it separate from the rest of the water molecule to do that, or is the whole water molecule going to attach itself? Or is that like a totally...
So the way it would work is that it's basically the whole molecule. So the molecule has this kind of structure, and it's going to orient itself to kind of minimize. It always reorients itself so that it can maximize the positive-negative interaction and minimize any positive or negative interactions. And then, and the other thing that I'm not showing here is that there's two orbitals that hold electrons, and those also hold some of the negative charge on them. So, to give some real-world examples of polar molecules, the most basic one is water. It's in the name. Hydrophilic, water-loving. Water itself is a hydrophilic molecule, because water easily is soluble in water. That seems obvious, but I mean it is a polar molecule. Ammonia is another one. The ammonia that you clean with is this ammonia gas that's been bubbled through water. And because it's polar, because the nitrogen absorbs the nitrogen concentrates the electrons that would be shared by all of them if it was a nonpolar molecule. That's what actually allows this ammonia gas to actually dissolve in water. Ethanol is another example. So ethanol, vodka, basically drinking alcohol. If you think about the way alcohol is usually packaged, it's packaged as a percentage of alcohol. So vodka would be about 40% alcohol. Like schnapps would be like 26% alcohol, something like that. The reason that it's a percentage is that ethanol is a polar molecule and it dissolves in water. Vinegar is another example of this. Vinegar, when you buy it, it usually says somewhere on the label 5% acidity. The 5% refers to the fact that you don't just have pure acetic acid, which is the vinegar. It's always cut with some water. And glucose is the other example. Glucose, like what you see in Pedialyte, Pediasure, basically sugar, a very simple sugar. And if you look at the 4 examples or the 5 examples that are shown here, there's something that they all have in common. Can anybody guess what that is?
They're all clear.
Yeah, they're all diluted with water. They actually can have different colors. It's not that they're clear. I guess they happen to be clear. I didn't realize that until you said it, but they're all diluted with water. And most of the time when you see a polar molecule, what you're going to see is that it can dissolve in water, and if you're using it for some sort of commercial purpose, you're probably going to cut it to get the concentration that you want, because the concentration is going to be tied to how strong or how weak it is. But that's one of the benefits of polar molecules, is that we can cut them with water. Water is usually very easily available. You wouldn't want pure acetic acid. Pure acetic acid would burn a hole right through whatever plate you put it on. 5% acetic acid can be put on a salad. Yeah.
If you have, is there a way to extract the more concentrated amounts? Like, can you undo the dilution?
You can. It's tricky with these molecules because of what I showed right here. Because of this charge separation, when this molecule is in solution, so the rubbing alcohol that you buy is also cut with water. It's also attracted to the water molecule. So the positive end lines up with the negative end, and the positive ends of the negative ends of the water line up with the positive end of the molecule, which means that the way that you would try to strengthen this, you would have to use the way that you would try to concentrate these molecules would be to use a distillation. So if you think about vodka is a pretty good example. So if you think about how vodka is made, first you ferment some potatoes or whatever it is you have, and you end up with something that probably has a concentration around like 12% alcohol, 12 to 10% alcohol, not very strong. And then you have to use a still. So the way that you would concentrate any of these is you would take the solution, so ethanol in this case, plus water at somewhere around 10%, and then you would heat it up. And
What does that say, sorry? H2O plus?
Oh, plus ETOH, sorry. You take the water plus ethanol, and you heat it up, and all of these molecules have different boiling points. So in principle, at some point you'll get more ethanol than you will water. But because they have such strong interactions, it's very difficult to get the pure ethanol to come out. To do that, you have to add another molecule that kind of absorbs the water into itself. You can push it up to about 96% if you really put a lot of effort into it. And you have a much more complicated setup than this. So if you were making moonshine, you'd probably get it at around like 40% at the most. Maybe like 50%.
I just make coffee.
So you can concentrate these.
Wouldn't it be easier just to add more acid to it?
It would be, but so
Instead of
going back to the
diluting it,
alcohol
yeah,
example.
she asked how you could break it
Yeah.
apart.
Some of these are harder to make. So if you're making alcohol, it's a lot harder, if not impossible, to make a pure alcohol solution out of nothing. It's a lot easier to just get some potatoes and let them sit and make like 10% and then distill it. So especially if you think about like a cost, if you think about the cost to make something from scratch versus the cost to to make something cheaply and then distill it. Usually it's done in most chemical plants to make it cheap, make a lot of it, and then purify it after the fact. Unless you have to be really careful. So now we can look at the flip side of this whole thing. We talked about polar molecules. So now let's talk about non-polar molecules. Molecules that essentially aren't going to have that kind of separation of charge between the two. So we can look back at this propane. Propane does not look like this, but
Yeah.
it's not really exciting to show a picture of nothing, which is what the gas would look like. So, but the grease that you get, grease that stains everything and gets on your hands, is essentially a variation on propane. It's a bigger molecule, but it's going to be another kind of non-polar molecule. Anything that's greasy or dirty is, I would say, almost always going to be a non-polar molecule. And if you think about how they interact with water, we talked about how the water tries to align the negative and the positive charges. The negatives attract to the positive, and the positives attract to the negative, and the hydrogens are going to point towards the oxygens. If we start sticking nonpolar molecules in between the waters, what you can see is that they can no longer interact with each other. You have this thing in the way, and this nonpolar thing doesn't interact with the water. It doesn't have any separation of charge to kind of let the water orient towards it. So what it starts doing is it starts pushing waters out of the way. If we look at examples of nonpolar molecules, we'll start to see what makes them nonpolar. So the first one, you'll notice it does have oxygens on it. So it does have these two electronegative elements on it, but because this end right here has such a long chain that does not have any electronegative molecules on it, this is what dominates the chemical character of this molecule. This molecule is nonpolar even though it has these two oxygens on it, because everything else in it is nonpolar. You can having an oxygen on it is not just an instant giveaway that it's going to be polar. You have to consider the rest of the molecule, and namely you have to consider the size of the molecule. If this molecule were smaller, if you only had this right here and not the rest of it, this would probably be a polar molecule. It would probably dissolve in water just fine. Because you have this long fatty chain here, that's what makes it nonpolar, and that's what makes it so that it won't become soluble in water and is soluble in other polar molecules. Beta carotene is another example, and you'll notice a lot of these are vitamins. Lutein is, it might have a vitamin name. I think it might be vitamin H, but I wouldn't write that down because I'm not 100% sure on that. But what it does is it gives eggs their yellow color. It also gives marigolds their yellow color. Beta carotene is another one. Beta carotene is what makes carrots orange. I'm sure a number of other chemicals are doing that too, but it looks a lot like this other molecule, except it doesn't have the oxygens here. So what that tells you is, when we talk about polar and nonpolar, it's not completely black or white. It's not that something is 100% nonpolar and 100% polar. Some things are more polar than others, and some things are more nonpolar than others. Water is very, very polar. Ethanol is almost as polar as water, but not as much. Isopropanol is probably going to be less polar than ethanol. And when we look at these two, they're both nonpolar, but the beta carotene is going to be more nonpolar than the lutein. The reason for that are these oxygens right here.
I have a question.
Yeah.
So, would this, I'm guessing it explains why vitamin E, beta carotene, and other non-polar molecules like those vitamins and stuff can be toxic if you take too much of them?
That's exactly it, is that.
They build up,
So if you think about
You can't pee it out.
yeah, so vitamin C is ascorbic acid, so it is polar. I don't know what the structure is off the top of my head, but it is polar, and that means if you take more of it than your body needs, it goes out through the urinary tract, and your body disposes of it. These vitamins here, they get stored in the fat cells. So they don't really have any place to go. You don't really have a waste system that can dispose of the fat as easily. So if you eat a lot of these molecules, if you ingest a lot of these molecules and they store up in your fat cells, and then all of a sudden you start losing weight very quickly, you start releasing all of them into your system very fast, and that's where the toxicity can come from. Yeah, that's why, I mean, so these are all fat soluble molecules, or fat soluble vitamins, and the fat soluble vitamins generally are more dangerous because of that. That doesn't mean like don't take them, just don't take like handfuls of them, but
Don't eat a bag of carrots every
that would probably be fine because the concentration of that would be
that's
probably
there.
less than the pill.
It's if you take it straight, if you take the
Yeah.
beta-carotene, or if you take the vitamin E jar. Because I do know some people who eat bags of carrots a day.
So if you eat a lot of beta carotene, what can happen, like people who drink carrot juice
Turn a
nonstop,
little orange in there.
they turn a bit orange.
Orange. My
That's
son
what I was
did.
gonna get to, because I've seen people turn orange.
The orange comes from, this is building up in the fat stores in the skin cells. And it builds up to a point that you can actually see it, which means you probably have too much.
Right. Would that be the same for lutein?
I'm not sure.
I heard it's good for your eyes.
It is good for your eyes, so I mean, I'm not a biologist, so I couldn't really tell you like where it's actually being stored up. I read that it's stored in your eyes. I don't know if it would cause your eyes to turn yellow or something like that. I mean, that's the color it's associated with, is this yellow color. But I don't know if that would actually happen. So. Vitamin E is another example, and you can see here, this is the dead giveaway for a fat-soluble vitamin, is the capsule. The reason it's in the capsule is because it has to be stored in oil. And if you want to store something in oil and you want someone to be able to take it, you put it in this little capsule here. And lastly, for a non-vitamin example, butane. So lighter fluid, essentially. Or the lighter fluid gas. The kind you would use for one of those high-pressure lighters. It's another non-polar molecule. It's smaller than these other ones. And because it's smaller, it's going to be a gas. I felt like I had to have a small one because all the other ones were huge. But I didn't want to give the impression that the size is what makes them non-polar. It's just the fact that there's not a concentration of electrons in one part of the molecule. So now we can start to think about what happens if we try to stick these molecules in water. We talked a little bit before how they block the waters from interacting with each other. And you'll notice the waters are all pointing in the same direction. That's only because it was easier to draw that way. In a real liquid, the waters would be twisted around and they'd be pointing so that they could align all the positive ends with the negative ends. And I tried to do that a little bit here, but not like you would see in a real system. So. But what you can see, let's imagine we took this thing and we stirred it up really well. So we have all of these non-polar molecules that are surrounded by water. What is going to eventually start to happen is that these waters are going to want to interact. They're not going to be able to interact with these polar molecules, but they are going to want to interact with the other waters. So as the waters start aggregating with each other, what ends up happening is they start pushing all of these non-polar molecules together. So now what eventually happens is the non-polar molecules are going to globulate together, and all the water molecules are going to be on the outside. If this were on the surface of something, so if you had a beaker, if this were on the surface of a beaker, what would happen is that this would be the bottom of the beaker and this would be the top, because these are going to be less dense than these. So if we think about this, though, this actually I think was covered in some of your other lectures, and it's one of the more important effects that you see when you mix together polar and nonpolar molecules. The fact that these nonpolar molecules aggregate with each other is what actually causes proteins to fold. I think with Aaron Dinner, you guys did a hands-on experiment where you tried to match up all of the white.
Yeah, we folded this.
Yeah. The reason that happens is because the white molecule, the white beads, would be similar to these hydrophobic molecules. They're hydrophobic residues. They're sticking out into the solution, and all of the water molecules can't interact with them, but they want to interact with the other molecules. So they form this kind of cage around the. They form this cage around the hydrophobic molecules, and what that does is it it causes the protein to hide those hydrophobic molecules on the inside so that they all glom together the same way that these hexane molecules glom together, and then the water or other polar molecules are on the outside and they surround the hydrophobic core.
I have a couple questions. I
Sure.
think. So this would be in a, is this in a water-based environment that
Yeah,
this
so this would
happens?
be like if I took these molecules and I threw them in a beaker of water and I put a lot of effort into mixing them together. Like if I were to use something like an ultrasound, which is going to use ultrasound waves to basically shake these into solution, and then I let it sit for a couple hours. What I'm eventually going to find is I'm going to find that these, the nonpolar, what used to look like it was one liquid will eventually separate again back into two. And what, yeah, so these are my examples of that. And I think the key thing to, the key expression that we usually use in chemistry is like dissolves like. And I think it's extremely important because you can understand a lot of biology by using this phrase. If we look at this salad dressing right here, somebody's taken that salad dressing, they've tried to mix it up because nobody wants to just pour the oil part onto their salad. And what you can see here, it's not like the green parts or the herbs are evenly distributed throughout the whole thing, but there's still a separation between the oil and the water. Different parts of the salad dressing want to hang out in different parts in the different layers. All of the herbs are hanging out here in the oil because the herbs are going to be more nonpolar, or at least the molecules on the outside of those herbs are going to be more nonpolar. And then you can see all of the things like the lime juice here, they sit in the water because the lime juice is going to be some sort of an acid, and acids are going to be, it's going to be like citrus acid and ascorbic acid, and those are all going to be polar. So they get dissolved in the water. Everything that's nonpolar gets dissolved in the oil, and the oil can't dissolve in the water, so it separates out. If we look at a leaf, we put some drops of water on the leaf, it doesn't get absorbed into the leaf, even though we know that on the inside of that leaf is water and all other water-soluble molecules. But the leaf is covered with a layer of wax. The wax is nonpolar, and that keeps the water from going inside of it. We look at a lava lamp. The lava lamp is filled with water, and then we have this colored oil here. And as we heat up the oil, it becomes less dense. It rises to the top, and then when it cools down, it becomes more dense, and it goes back to the bottom. But these two never mix. If you take the lava lamp and you try to shake it again and again, you'll get smaller and smaller globules, but it doesn't suddenly become one color. The reason, once again, is like dissolves like. Polar things dissolve polar things. Non-polar things dissolve non-polar things. And they don't mix unless you put in a lot of effort to mix. Not a lot of effort, but you have to do something to them to mix them.
You have to change it, right?
No, what you can do is you can sequester them. And so that's what this is right here. So I talked about polar and non-polar. So I will answer your question, but the next slide right after this answers that question exactly. And what does that is an amphiphile. So an amphiphile is essentially a polar and non-polar hybrid. It's a molecule that does have one region that's definitely polar. You have this head group right here, and that head group attracts a lot of electrons to it. But because the rest of the molecule is so long, you also have a polar part. So when I showed this example right here, I said that this molecule was polar overall, and the reason for that is that these OH groups here, they don't really pull that many electrons towards each other. If it were a smaller molecule, it would pull in enough to become polar, but they're not the most polar chemical groups here. These are very polar, all of these. They have 2 oxygens, 3 oxygens each, plus the sulfurs, which also pull more molecules towards themselves. So you have a lot of charge that's being held here, but then as you move further and further away from that head group, because the tail is so long, you're going to have a region that's going to be nonpolar. So now you have a molecule that can do both things. And what it can be we can use it for, and what we usually use it for, is to dissolve polar things in nonpolar liquids, or vice versa. And soap is the best example I can give of this. So this molecule here, I don't know what its chemical name is, but it's essentially every soap that you've ever used probably has a molecule that looks just like this. Maybe there's some other stuff sticking off of it, but in general it's going to look exactly like this. You have this very polar head group here, and then you have this nonpolar tail here. So if we want to clean something, you can imagine we have dirty dishes or something like that. We wash them with water, and that's going to get some of the stuff off, but it's not going to get everything off. The water's going to take off everything that dissolves in water. Everything that's polar and sitting on the plate will get washed off when you just wash the dish off. Anything that's greasy and nonpolar, chunks of food, etc., is going to stick to that plate. And the way we can get rid of it, we can wash it with more water, and it's not really going to make the plate clean. But if we stick this soap molecule on it, what ends up happening is that the soap molecules are going to surround the piece of nonpolar junk, and the head groups are going to orient towards the solution, and this nonpolar tail is going to orient itself towards the nonpolar piece of junk. Now, if you think about this, would you say that this whole thing is going to be soluble in water or not soluble?
As a whole unit,
Yeah.
as a whole unit, if it stays together, it's
Because the outside of the heads are positive of going toward the water.
Exactly, and that's exactly what happens. This is why soap can actually wash grease off of plates. Is it surrounds all of the grease molecules, and it dissolves the grease molecules in the nonpolar part, and then the whole thing becomes dissolved in the water and gets washed away. So this is still another example of like dissolves like, but the molecule is serving two different functions. The molecule has two different chemical characters to it, and each of them is performing a different function for this. Yeah.
So I didn't hear some of that up there, but I'm assuming you said that, like the part that has a little circle on it, that's the polar,
Yeah.
and the non-polar is on the inside.
Yeah, exactly.
doing its work. Okay.
And so now if we go back to the salad dressing example, when you buy salad dressing in the store, some of it's separated, but some of it's not separated. You can shake it up, and it'll stay as kind of a homogeneous liquid for a pretty decent amount of time. And the reason for that is another example of this, is that it forms an emulsion. So once again, you have some sort of molecule in the salad dressing, I think in general it's mustard seed, that is going to have some sort of polar head group, some sort of nonpolar tail group, and they're going to surround the oil, and they're going to dissolve it in the liquid. Milk is another example of this. So I guess I didn't define emulsion, but emulsion is going to be a liquid that's a combination of a liquid that's going to have polar molecules dissolved in a nonpolar base, or vice versa. And milk is probably the most common example of this. We know that if we buy a whole milk and we shake it up a lot, we can make butter out of it. We can separate out the fat from the milk itself. But then if you ask the question, now that we know that the fat shouldn't really dissolve in the water at all, how is the fat actually in this liquid milk? The milk is mostly water, but the fat is somehow dissolved in the water. And the way that happens is that there's molecules in the milk that are going to have a polar head group and some sort of nonpolar tail, and they store all of the butter fat in these kind of little micelle vesicles. This would be called the micelle, and they're going to store the butter fat in these kinds of structures. When you shake up the milk, you start mixing all of these up, and you start breaking open some of these vesicles, and then the butter fat can globulate onto and form bigger and bigger chunks of butter fat, and eventually it's going to separate out from the solution. There's not going to be enough of these molecules to be able to cover up all of the butter fat and make it soluble in the water.
So that's how you get your non-fat milk.
Yeah, that's it. Exactly it. Is you add some sort of force to it to break these up, and then you separate out the milk. I think that's you can buy the milk that hasn't been made homogeneous that has the cream at the top too, still.
Mhm.
They still sell that, and then if you shake it up, it sort of dissolves, but not really that much, because you need enough of these molecules to really make the whole thing kind of the milk that you buy at the store in the plastic gallon.
So the same principle would go with making whipped cream?
Whipped cream is a little bit different.
A little different?
Whipped cream, you have proteins there that are helping to build that structure, and they're trapping air inside of them. That's why if you were to, the foam that you see in milk, if you were to shake the milk up and you see a foam, like in a milkshake and the foam stays steady, or in beer is another example, that's actually a matrix of proteins with
The
air, like a kind
The
of net
foam
of
part
protein.
of it is
Yeah,
a
the foam
matrix?
is.
Oh,
So beer foam forms because you have a lot of protein in beer. Milk foam forms because you have a lot of protein in milk. If you take a soda and you shake it up, it'll be foamy for a little bit, and then it dies down because there's no protein in the soda.
This is so cool.
can we get access to your place?
So what we've done here, everything we've done so far is we've built up a toolbox. I would say a chemical toolbox to understand lipids in biological systems. We talked about what it means for a molecule to be polar, what it means for a molecule to be nonpolar, how things can dissolve in water, which things would dissolve in water, which things don't dissolve in water, and how we can kind of trick the system and dissolve something in oil or water that doesn't want to actually dissolve in that with these amplifiers. So now I think we have everything we need that we can actually talk about lipids. And essentially, lipids are fats. Every natural fat that you ever come in contact with is essentially a lipid. Some things are fats that you don't think of as fats.
Wax is a fat.
Beeswax is a fat. It's a lipid. These structures don't correspond to these structures, but these are just three examples of, these are chemical examples of lipids, and these are just picture everyday examples of lipids. Showing you a picture of cholesterol is not really exciting. It looks like a white powder. A free fatty acid is not exciting either. The triglyceride is olive oil essentially, and the butter too. Depending on, there's some little variations in the chemical structures that make one look like butter and make one look like olive oil, but this is the one that corresponds to the pictures. The beeswax looks different, but in general, when you think about a lipid, what you're really thinking about is something that's non-polar or something that has a big non-polar part to it. That's essentially an amphiphile. So if we look at these examples, we have cholesterol. Cholesterol is mostly non-polar. It has this little oxygen right here, but it's not really so electronegative that it's going to pull all the electrons towards itself. So the environments that cholesterol is going to want to be in are going to be fatty type environments. It's going to be dissolved in other lipids, in other nonpolar molecules. We have this fatty acid here also. It's a bit more of an amphiphile than the cholesterol is, but it's still probably not, this is not so electronegative that it's going to be a true amphiphile that could form a micelle. It's still probably going to be stored mostly in it's still mostly going to dissolve in other fats. We have this triglyceride. You can see there's kind of a trend here. These head groups are not so nonpolar, are not so polar that they turn this thing into an amphiphile. This is also going to be mostly a nonpolar molecule, which is what you would expect. The triglyceride is olive oil, and olive oil you think of as a pretty standard oil. You don't think of it as being like a detergent. You wouldn't clean something with olive oil.
Quick question,
Yep.
is that all vegetable oils or just olive oil?
All vegetable oils are
All
lipids, and they're all going to have
vegetable oils.
similar structures to
Okay.
this.
And so that's.
Big, long, fatty tail.
You refer to that like butter too, right?
Butter would be the same thing too.
Okay.
I have a question.
Maybe it'll, yeah, go ahead.
I should have asked this
No,
earlier.
no, no.
The long tail, each bend, what is the tail made up of? Because you've always been saying oxygen or hydrogen
Yeah, so
and
the tail
carbon.
is-
What is the tail actually?
The tail is shorthand for, the way the tail is drawn there, I should have covered this at the beginning, is shorthand for carbon hydrogen structures. So this would be,
Okay, so it is, okay. Thank you.
So whenever you just see the lines,
Mm-hmm.
the lines essentially mean carbon bonded to another carbon with a single bond, and then as many hydrogens as you need to fill up the octet.
Okay.
So this one is only bonded to one carbon, so it has 3 hydrogens on the end. This carbon is bonded to this carbon and this carbon, so it needs 2 more hydrogens to bond the 4 things. And so then if you were to see something like this,
That's that double line right there.
yeah, so that double line corresponds to this double line.
You show it in the triglyceride and the fatty acid.
So if I draw this structure right here, so I have the double bond, I think I just showed the bad organic chemistry. I haven't done organic chemistry in a long time. Either way, it looks like this. Whenever you see the double bond, now this carbon has 4 bonds on it. It has the 2 hydrogens and these 2 bonds right here.
So they're sharing, the
They should
carbons
not
are
be here.
sharing, they're not sharing, but,
Well, they have a bond, so they're bonded to each other. So there's a double bond. Carbon can have single bonds, double bonds, and triple bonds. Triple bonds, I don't think you see much. Carbon triple bond without a nitrogen, you wouldn't see much in biological systems, but you will see these double bonds. So the single bonds are more flexible. The double bonds are not very flexible. So if we, and actually we can talk about this in terms of the lipids too. If we think about these, in general, these 2 halves can rotate freely. There's some energy required to rotate, but
Each of them are
not too
separate,
much.
is a separate.
Yeah, so this region and this region, in a real system, they would rotate. They'd be able to rotate. This double bond, it takes a lot of energy to actually cause that to rotate. And it's because of the way that the orbitals are set up with respect to each other. But to cause these 2 hydrogens to flip is going to take a whole lot of energy, and it's going to take a lot more energy than you would find in a biological system. So if we look at these molecules right here, these single bonds can rotate freely and they can become disordered. And these double bonds are going to be, they're not really going to rotate, which means that this structure here can move around and become more disordered. This structure and this part can become disordered, but they're going to have to, with respect to each other, it's not going to be flexible here at this bond. This is not going to be flexible here at that bond.
are double bonds considered to be either polar or non-polar?
It would depend what's bonded to them. So if you had an oxygen here, that would pull the electrons towards itself. The polarity is always going to be a function of how many electrons can be shifted on one part of the molecule. So this region here would be more polar, but out here, as a whole, this molecule is not really going to be polar at all. The example though, so all of these here really are amphiphiles. You can essentially think of them as very nonpolar molecules. This last example here on the bottom, this molecule is definitely an amphiphile. So this is a phospholipid, and you can see here it has this phosphorus with 4 oxygens around it, and this nitrogen group here, and this end right here is going to be very soluble in water. This is a very polar end group, but these tails right here are not going to be polar at all. So if you start to think about what we talked about with the soap before, these phospholipids are going to act a lot like the soap molecules. And what that's important for is going to be when we think about the cell membrane. So what the cell membrane is made of lipids. We can look at this very complicated picture, and there's a whole lot going on here. But we don't really need to understand all of that. What we need to understand is these molecules right here. Each of these are individual phospholipids. They're going to look like this. So the white head group, the white ball on the top, that's going to be this region right here, this polar region. And then this this orange part here is this fatty tail right here. And then this is going to be inside of a cell, so it's going to be surrounded by water. So here's a picture of a real cell. This black here is the cell membrane. This is the nucleus, and the nucleus also has its own membrane around it. And those membranes are made of lipids, and they're made of those phospholipids specifically, and there's cholesterol and a bunch of other stuff in there. And we can understand it. Oh, this is bad grammar, sorry, I forgot to finish that sentence. I meant to say it can be understood in terms of this like dissolves like concept. If we look at a picture of these phospholipids, so these are just any two phospholipids that I pick out of a hat. We can, once again, they have this polar region right here, so that's all of this, and then they have this non-polar region right here, and that's all of this tail here. And if we were to put them in solution and shake them up, what's going to happen is they're going to orient with the tails towards each other, and the the head groups towards the water. And when that happens, what you can see is they start to form this kind of a structure here. This, what we call a bilayer.
What is it called?
A bilayer? A lipid bilayer. And this goes back again to this concept of like dissolves like. The nonpolar head group, the nonpolar, that should be tails. Tails point towards each other. And you can think of these tails as a liquid. The tails are solvating each other, and then the head groups sit in the water. And what you end up doing is you end up forming a barrier for the cell. If you think about why cells have a cell membrane at all, what they really want to do is they want to separate whatever's happening inside of the cell from whatever's happening outside of the cell. You have a lot of important chemistry that you're trying to control. And if you have this chemistry happening without some sort of a barrier that you can't control what goes in and what goes out of it, you're suddenly going to find that your reactions aren't going to go the way you want them to go. And that's what a cell is doing. A cell is essentially a chemical factory, and it's trying to control very specific chemical reactions. And the way it protects itself is it creates this bilayer. It makes this molecule, and this molecule has this polar head group and this non-polar tail group. And the tail groups, because this molecule is an amphiphile, the tail groups orient themselves to each other and essentially dissolve one tail into another, and then the head groups stick out to the top, and you have this cell that is soluble in water but doesn't let anything pass through it, because if something wants to get through this cell, it's going to have to be able to, it's either going to have to be very, very small, or it's going to have to be able to somehow be soluble. It's going to somehow be able to get through this polar head group, so it'll have to be polar, and then somehow it'll have to be non-polar in the middle. So how do things get inside of the cell? The cell has proteins that control all of this. One of the ways that things get inside of the cell membrane, you'll have some sort of protein that sits in the cell membrane, and it can control what goes in and out. And there's a lot of other membrane proteins that do a lot of other functions. And if you think about how does this protein live inside of the cell membrane, the way it's going to do that is it's going to the part that's inside of that lives with the tails is going to have to be non-polar. So you guys did the protein study before, where you guys did the protein example before, where you tried to line up all of the white beads, because you know that hydrophobic residues aren't going to want to sit in solution. A protein that lives in the membrane, any part that's in the inside of the membrane in this nonpolar region, is going to have to do the opposite. It's going to have all of the nonpolar residues sticking out into the membrane, or else it's not going to be able to be dissolved by these lipid tails. It's not going to be able to live there. You're not going to have a favorable interaction between these two tails. And the reason, once again, is like dissolves like. Anything that sits outside of the membrane into the water is going to have to be polar. So you have a lot of proteins that have these polar regions on the outside with the hydrophobic core on the inside for the part that's sticking in solution, and the part that lives in the membrane has to be non-polar on the outside. And if you want to use that protein to have water go through it, then you're probably going to have some polar residues on the inside too. I know that's a lot, but really, I think once again we can just think if we have a cell membrane, the key thing to get from this is, because like dissolves like, you can create this very complicated cell membrane structure and understand it in terms of this basic idea that the nonpolar parts dissolve the nonpolar parts, and the polar parts live in the water, and they actually allow the whole cell to be soluble, because the cell has to be able to live in water too, because people are mostly water. And if we look at if we look at a more chemically accurate picture of what this looks like, so this is using VMD. What you can see here is I've colored all of the head groups, or all the polar groups, with this green and red color. There's two different kinds of lipids, so one is red, one is green. And you can see how the tails look very disordered here on the inside. And the reason for that is that you can actually think about this membrane as a two-dimensional liquid. These tails are considered to be in a liquid state. Even though the membrane definitely has a structure to it, we still consider these tails are disordered the way you would think of a liquid as being really disordered. And you can see right here, just to kind of show the disorder, I've colored every tail a different color, and so you can see here that it's definitely very mixed together. And the other thing is, that I want to mention too is that if you look in this picture right here, this is a view from the top. It's not like it's all green and red. You can see holes in it. Thanks. You can see holes in it. Some of the membrane is actually, or some of the hydrophobic parts are exposed to the water. And that's why small molecules can actually get through this. Water is sometimes found inside of here, but it's not going to be a lot of water. It's going to be like one molecule, and maybe it'll find its way out again. But it's not a perfect seal. There's definitely holes in it, because stuff still has to get in and out of this cell, and it still has to be flexible. It can't just be a solid rock.
When you say it dissolves each other,
Dissolves.
does that mean it starts to disappear and dissipate?
No. It's when I say dissolve, I mean that it becomes more like a liquid. So they're interacting strongly.
Oh, okay.
So I'm using that more like an analogy.
Quick question, how
Sure.
thick is that?
It's a cell membrane, right?
Yeah, how thick is the membrane?
I would say somewhere about. What would you say, Joe?
It's like about 40 angstroms.
That's right. Yeah, 40 angstroms. So that would be.
An angstrom member is about the size of a hydrogen atom, so it's basically like 40.
45
Yeah.
So 10 to the. Yeah, 40 times 10 to the minus 10.
So what
meters.
type of microscope did you use to see something not necessarily, like, well,
So
not on this scale, I mean, that would be an electron microscope, isn't it?
So electron microscope would give you a picture like this,
Okay.
and you can actually. I think you can get better now. This would be another electron microscope picture, but I mean you can. I mean you might have. If you think back to like a high school biology class,
Yeah.
you can see the membrane. You don't see a lot of detail, but you can see the membrane of just like an onion skin cell. When you see those squares, the outside of the square,
The inside of the celery.
the outside of the square looks like this. And chemically, it looks like this. So it's, you don't see any of the detail. I mean, you just see like a black line, if I remember what it looks like.
And one more question, so compared to the stuff that's inside, is this thick? Compared to the
No,
stuff.
this is like a thin kind of like skin on the
You mean
outside of the cell.
inside of the cell?
Compared to the chemistry
Contents.
thing that's going on inside.
To the entire, so if you think about the cell, this would be like a very thin skin on the cell, and then the cell is gonna be big or large depending on what kind of cell it is. But the cell is much bigger than the thickness of this.
Is it like, could you equate it to that, that membrane like when you crack an egg?
So it looks like it,
Using
but it's not actually
analogy.
that egg is not a cell. It looks like a cell, so it's a good kind of model of a cell,
Yeah, right,
but it's
yeah.
not, but it wouldn't be that. What is that?
But as just sort
Yeah.
of a proportional comparison?
Yeah, I mean, they have some of the same parts, but I think the egg doesn't have like a good skin on it though. I guess it has that
You know when
little
you-
skin on the shell, right?
Yeah, like when you hard boil it. and
Yeah,
then it
yeah,
comes
yeah.
with
I mean, I think that would be a fine model. I remember doing that as a kid and cracking open the egg and looking at it. But
just in reference to the membrane, I guess.
yeah. So they have those parts. You can also think of it like, if we're gonna use an analogy like that, also, I mean, like the skin on like a peach or something like that would be another one.
Mm-hmm.
It's basically a skin on the cell.
So inside the cell, would there be molecules that would be larger than 40 inches on the inside of it?
There'd be proteins, I think. Yeah, there could be.
Yeah.
Yeah, that's definitely so. I mean, this is the inside of the cell. So this is the membrane right here. And this would be a very, very small cell. I don't think cells would actually be this kind of size, but this is the nucleus and it has its own membrane. And there's all of the genetic
Which
stuff
is
on the
made
inside.
up of the same type of
Made up of the same
membrane
kind of
that the outside,
phospholipid.
okay,
There's different kinds of phospholipids and different proteins, and cholesterol's gonna be
Right.
there.
okay,
But the basic concept is always gonna be something that looks like this.
okay.
This, if we think about this as if we try to think about this in the simplest terms possible, this is the picture that I would say you can think of for a cell membrane. Everyone knows there's a lot more going on. All of these would be different kinds of molecules, and there'd be there'd be some cholesterol which is smaller and lives mostly here, and all of that will affect the structure, but the basic picture is always gonna be some sort of amphipathic molecule that lines up to form this structure. So big things do need to enter the cell. We know that they need to enter the cell. So then the question is, how do big things get inside the cell? They can't pass through the membrane,
Yeah, they do, they could help.
so they actually form.
Don't they get help passing through the membrane?
They do get help, and the way that they get the help is by forming this vesicle, by forming this pocket. Something comes in, and then a
tells it
lot of
to.
proteins work in a very complicated way to absorb whatever's coming inside in a new little cell membrane. So suddenly, whatever object you want to get inside of the cell gets wrapped up in a kind of smaller, in its own little membrane vesicle is what we call it. And now it's in this kind of pocket. So this right here would be a membrane vesicle. And then the cell uses a whole bunch of other machinery to get it where it needs to go. So in the picture here, something on the outside of the cell needs to get inside of the nucleus. It's way too big to fit through the cell membrane. Only small molecules can really do that. So what happens instead is the cell membrane structures itself around the stuff that needs to get inside and forms this vesicle. And then the vesicle
It breaks off.
breaks off, and then it gets transported to the nucleus, and then it gets reabsorbed. Because the nucleus the nucleus is also going to be made up of this, is also going to be surrounded by this membrane vesicle.
So then it creeps in.
Does that work in reverse as well?
Yeah,
To-
stuff also
to
gets
get
sent out.
waste stuff out? Okay.
So this would be, going inside of the cell would be called endocytosis. Going outside of the cell would be exocytosis.
Endo coming in.
Endo going in, exo coming out. And there's another interesting thing here too, is that, so I said before, why does the cell have a membrane at all? The cell has a membrane because it has a whole lot of functions that it wants to perform, and it doesn't want anything interfering with that. Why does the nucleus have a membrane? The nucleus has a membrane because it also has a lot of functions that it doesn't want the rest of the cell interfering with. Different cellular organelles also are going to be surrounded in different membranes, and whenever there's a membrane in a cell or in a biological system, what it's essentially doing is it's keeping stuff out and keeping stuff in, and somehow it gives the cell or the nucleus or the organelle some sort of control over what's happening. And if you think about things that are alive, there's a whole lot of really complicated stuff happening. And if you just let everything interfere with everything else, stuff will probably go wrong. And those are the kinds of problems that lead to diseases, to lead to genetic diseases, etc. And the more that the cell can control exactly what's happening, the better the cell is going to function. So it's a very complicated system, and the kind of big evolutionary advantage that cells have is that they have this membrane that controls all of the chemistry. That's the difference between random biochemical reactions happening in a pond and something that's actually a cell or a bacteria, is this cell membrane. I think, so I'm ending here, and then we'll do some activities that play around with this idea of lipid. But really, I think the take-home message I wanted from all of this is that this is a complicated structure. This is very difficult to understand, and frankly, people don't completely understand it. This is a pretty easy system. It's a system that you understand, at least intuitively. If you made salad dressing, you know that some parts dissolve in the oil, some parts dissolve in the water, and then you have to somehow mix it up together. And if you want this thing to be one liquid, then you're probably going to have to add mustard or something to it. The same, all of the chemistry that we use to understand the salad dressing, we can then apply to the cell, and we can use it to understand how the cell is able to keep stuff on the outside, stuff on the inside, and actually control that process.
Why does mustard work?
Because it has an amphiphile in the mustard seed. It has some sort of molecule in it that's an amphiphile. So then it starts to dissolve the oil in those micelles.
That's funny because you see that in.
Or actually, it'd be vice versa. It starts to dissolve the water in the oil. In the polar part surrounds the water molecules, and then it dissolves them in the oil because you have a lot more oil than water in salad dressing. I think I got this from some sort of health site. That's why it has so little oil. Usually it should be like 4 times as much oil. But it was a pretty picture. So. But that's why if you look at a vinaigrette recipe, Dijon mustard is always one of the ingredients.
Mustard powder, I see that too. And because the mustard powder doesn't really add a lot of flavor.
Let's say, I
And it's
always
not
thought
a
it was
lot, it's
for
just
subtle
enough to
flavor.
dissolve one and the other.
Are the leaves
Very
and the
cool.
top, is that a weight thing? Or is that because?
It won't be density actually, because so I'm gonna conjecture here now, but I think it's because the leaves are surrounded in some sort of a wax.
Okay.
So the picture of the leaf I showed you before,
Mm-hmm.
so that means that the leaves are gonna be more non-polar. So they sit there in the oil.
Which is non-polar.
The reason that the water is not clear is because it's not water, it's lemon juice. So all of the stuff from the lemon, which is water soluble, is gonna live in the water part. In the polar part, all of the stuff all of the leaves and all the herbs and all of that's going to live in the non-polar part. And I'm not sure why the red is in the middle. I mean, I don't know if they mix it up or not.
You wanted us to see that
It's vinegar.
But yeah.
It's something that's lighter than water,
Hold on.
If it's cayenne pepper, it might just be. I can't explain the red, but it might just be that they didn't mix it good too. So.
But it's a good example of separating.
It is a really good example. I always thought of just oil and water, and so this example, it changes it now. So now it's polar and non-polar instead of
Yeah, exactly.
oil and water.
I mean, it still is oil and water, but why is the oil? What makes the
Right.
oil and oil, and what makes the water
Right.
and water? And that very complicated biological systems. They're still made up of just molecules, and the molecules can be understood in terms of this. Everything else is just details, but this is the big picture, I would say.
I like your big picture.
Yeah, good
Me
picture.
too. It's perfect.
All right, so